Hey guys, welcome to the disease unit lesson 7 lesson overview. This was exciting because I finally got my life together and gave you guys a checklist because I think that's going to help us. And I also got us started on Nearpod. Hopefully you guys are enjoying the Nearpod. I've been enjoying watching what you guys are doing, listening to your Flipgrid stuff because that's been super fun for me too. If you haven't gone through the Flipgrid stuff, initially I put it all on the end of the Nearpod. Later on, I just decided to make it a whole separate thing. So I think that was an improvement too. Just remember, I'm learning as I go, just like you guys are. Another thing, hopefully, especially if you know that like reading isn't your thing, hopefully you got into this immersive reader thing, which I think is really cool because it will like read things to you. It will help you like take a look at all of the words and parts of speech. You can do comprehension stuff. You can do all kinds of different text spacing. If you struggle with discerning between letters, you can make this text bigger. You can put it on a weird yellow background, whatever you want to kind of make it easier to digest. Okay, and then we're going to go into talking about Gila. All right, so Henrietta Lacks was a really cool lady. She was alive in the 1950s and died of cervical cancer. And they took part of her tumor and those are the immortal cells. So these are the cells that are still alive today, and they're called HeLa for Henrietta Lacks. Okay, let me put my purple on so you can discern between what I wrote before and what I'm writing now. All right, so earlier in this unit, we reviewed 4 aspects of cancer cells. Okay, that's what we did on like the second day of the unit that separate them from a normal cell. So we're going to read each piece and then we're going to talk about the different, we're going to kind of compare and contrast. Now, if you notice, there are multiple answers that you can put here. Okay, there's even 3 over here on excerpt number 3. There's just 1 here on excerpt number 4, and there's just 1 here on excerpt number 2. But anything else, as long as you can tell me about what you think, as long as you can compare and contrast our HeLa cells with our normal cells adequately, then I don't care what you put up here. Okay, a lot of us think that we have to have 1 from each, and that's not true. It's just a matter of what can you explain, what can you prove. Okay, then you're going to go to the next section, and this is going to be matching it with the pictures. Okay, so this picture, I said you could connect it to 1, 3, or 4 because it's showing how normal cells have division, and then whenever we have a damaged cell, it dies. But cancer cells, we have a bunch of these damaged cells, see, because they look the same, but they don't go through apoptosis. So I said that connected to number 1, okay, because it says it can divide indefinitely. It can go with number 3, okay, because it's talking about apoptosis. And then number 4, because again, it's super growing out of control. Okay, so for this one, then you just need to take your evidence, and the evidence can be from the picture here or from the article that you read before. And then your reasoning, remember, that connects the science. Okay, so why does this make sense? Picture 2, again, we've got cell suicide or apoptosis. We've got uncontrolled growth in cancer. Again, we can use evidence from the picture or article. And then reasoning is the science behind it. And then we've got number 3. Number 3 doesn't really have any options. It's really only connected to excerpt number 2, which is why I kept it here. And then number 4 connects to 1, 3, and 4 again. So it's just a matter of what you want. But here's an example for you. I said the evidence specifically cites the tumor suppressor gene is P53. And over here, it says that the tumor suppressor gene is mutated. So if I have a mutant P53 gene, then I'm going to have loss of growth control or an uncontrollable tumor. For our vocabulary, we just have 4 words again. P53, the super special gene. Telomerase, which is a part of the telomeres. HeLa, and HPV. All right, and then in the WURQ, or the work, we've got our big question: how do genes help regulate the cell cycle? Who was Henrietta Lacks? She was a woman who died in the 1950s of cervical cancer, and her cells are immortal today. So that might be something I add. Her cells are still alive today. And I might even add her cancer cells. Be a little more specific there. All right. Why is she important to the history of science, especially medicine? Her tumor cells are mutated, so they are the only cells on Earth that are immortal, or they divide forever. And then write down one more thing you figured out today. What questions do we still have? Why don't Henrietta's children have cancer? Why don't her children have immortal cells? And then write down one more thing you learned from that lesson. And then we've got, why are Henrietta's HeLa cells so special? What makes them unique? And I said they have a mutated p53 gene and a mutated telomerase. And so they have high tumor growth rate, and she has immortal cells. Then what were some of the ethical issues around using Henrietta's cells for research without telling her family? I said that it emphasized civil rights issues of that time, and it begs the question, who owns your cells? So think about that. All right, that's all I've got for today, and I'll see you next time.